mutations can appear in all parts of the viral genome. However, those that happen in the spike protein that is responsible for viral attachment and entry in the cell are of particular interest because they may have implications on the viral transmissibility, morbidity, and mortality, the ability to evade detection by diagnostic tests, or ability to evade natural immunity caused by infections and reinfection, therapeutics, neutralizing antibodies, or vaccines that target this protein. So, by sampling and sequencing viral genomes over time and space, we are able to identify mutations that occur in the viral genome. So there are currently more than 2 million SARS-CoV-2 sequences publicly available in GSAID and GenBank repositories. And these data are crucial to be able to track the evolution of the virus in real time. For instance, NextStrain is a platform that allows one to quickly explore these evolutionary dynamics without having to deal with the huge data sets of genomic data. Here on the left, we see a phylogenetic tree of SARS-CoV-2, where the circles represent sequences collected in different continents. And as you can see, we observe some degree of intermingling with sequences from different locations clustering together, which is a testament to being a human pathogen that travels along distances, long distances, due to human mobility patterns. So the pandemic started with viruses that were part of lineage A, shown in purple here in the phylogenetic tree, also called by another nomenclature system as clade 19. However, in early 2020, we observed that viruses with the mutation from D aspartate to G glycine in position 614 of the viral genome started to also circulate and increasing its frequency to later become the dominant circulating variant denominated B1 lineage or clade 20, depicted in green in the phylogenetic tree. Further studies demonstrated that the success of lineage B1 was due to the moderate increase in transmissibility conferred by the D614 gene mutation. So in one of our works, we demonstrated how by early March 2020, the B1 lineage had already been introduced and dominated the number of cases detected in the DC area in the USA due to introductions mostly coming from Europe. So the B1 lineage, as I was mentioning, was responsible by the initial wave worldwide. However, mitigation efforts, including decrease in travel between and within countries, led to the emergence of regional clades where different countries and more largely continents were characterized by their own viral diversity. However, summer ended and a new type of virus started to emerge called variants of interest or variants of concern. So since viruses are constantly going through the replication process of copying their genomes, it's normal for them to change over time. And as I've mentioned, viruses with different sets of mutations are called variants. Most of the time, these mutations are harmless or even make the virus weaker, and they quietly disappear without making any noticeable difference. But other times, a series of mutations occur that give the virus a slight edge over us. Some of these mutations confer different attributes, some of which have implications to public health, and thus are named variants of interest or of concern. These variants have mutations for which there is evidence that they confer a higher transmission rate or are coupled with increased disease severity, which can be reflected in the increase in the number of cases hospitalizations, or deaths. Variants of concern have also been shown to be associated with reduction in neutralization by antibodies generated during previous infection, reduced effectiveness of treatments or vaccines, or diagnostic detection failures. And so, due to all these factors, variants of concern might require more strict public health actions in an effort to control their spread, such as increased testing and lockdown enforcement. So what we have now around the world is this escalation in the classification of variants based on this ladder. So when we think, and starting from the bottom, when we think about horizon scanning, we think about surveillance of mutations and variants as they arise. And so one is looking for mutations that could be associated with reduction in neutralization by antibodies, or for mutations that we know are already present in other variants of concern. So your horizon scanning is looking for signals of concern. One might also see a rapid spread in a community or other settings, and that would move a variant to the category of variant of interest. So at this stage of variant of interest, one has evidence from genetic information that there is something different about this virus and that it appears to be spreading in the community. There are various possibilities about spreading. It can either be caused by the biology of the virus that makes it more transmissible, or due to a founder effect, where a virus goes into a population and appears to be more transmissible, but the spread is due to chance. The interesting and difficult question is when we go into variants of concern. So several variants of concern have emerged since late 2020, which I'll talk a bit more about later. But the distinction between variant of interest, or VOI, and variant of concern, or VOC, is very important because we might behave differently depending on which we are dealing with. The response to a variant of concern, or VOC, is usually accompanied by surge testing and greater public health interventions for control. And that comes to a gradual increase of evidence. And I say gradual because we need to generate that evidence. So we need an increase in the level of evidence that they have characteristics of increased transmissibility, immune escape, or lethality. And the final category is variant of high consequence. And we haven't seen yet any variant of high consequence. But if we think about a threshold of what that would mean, is that there is clear evidence that control measures have significantly reduced effectiveness relative to other circulating variants. So in late 2020, the UK reported the circulation of a variant that carried the mutation in position 501 along with other mutations. And of course, these were concerning news as it looked like these mutations confer substantial increase in viral transmissibility. Shortly after, other lineages sharing the same mutation on position 501, along with varied mutation constellations, started being reported in South Africa and in Brazil. The N501Y mutation arose independently in several variants of concern, including the B117, the B1351, and the P1 lineages. This mutation is near the tip of the coronavirus, as you can see here, where it seems to change the shape of the protein to be a tighter fit with human cells. But besides their increase in infectiousness, these other variants that carry a mutation in position 484 in the viral genome also appear to be less susceptible to some vaccines or our immune system. The E484K mutation arose independently in multiple lineages, including the B1351 and the P1 lineage. And this mutation, again, as you can see, occurs near the top of the coronavirus spike, where it alters the shape of the protein. And this change may help the spikes evade some types of antibodies, as at least one trial has shown. So, genomic epidemiology efforts like those led by the Next Strain team provide enormous help on identifying and tracing the spatial history of such mutations. In this phylogenetic tree, the teal dots represent the viral circulating diversity, whereas the yellow, blue, green, and orange dots represent the variants firstly detected in the UK, South Africa, and in Brazil. These have been classified as lineages and are now referred as B117 or alpha, B1351 or beta, and P1 or gamma variants, respectively. To note is that despite sharing mutations, these lineages appear to have emerged in different locations in the world, and after meticulous phylogenetic analysis, this is proved by forming different clades in the tree, as you can see here. Let's see if you can see my mouse. So as you can see by the squared lineages. And so this suggests that there is such fitness advantages to these mutations that they were generated and fixed independently through distinct evolutionary pathways. Another commonality across these variants is that they appear to be evolving faster, meaning that they accumulate mutations faster than their ancestral lineages and those currently co-circulating. And when studying the evolutionary history of pathogens, it is important to estimate the rate at which they accumulate mutations, as this gives us an indication of how soon control strategies like vaccines need to be updated. And so here in this plot, we can see that the initial lineages of SARS-CoV-2 accumulated mutations slowly at rates comparable to that of seasonal coronaviruses, as you can see by the blue dots. However, the recent variants, the red dots, appear to be acquiring mutations at rates even higher than those of influenza A subtype H3N2 viruses, for which we need to update vaccines every year. So now I want to talk for a moment about what drives the selection of variants of concern. And this is a very important question because we want to be able to identify and try to stop it if we can. So as we continue to trace the origins and evolution of these variants, we question about the mechanisms that might have led to their emergence. And of course, it is reasonable to assume that in places with high infection rates, there is more opportunity for viruses to generate mutations that might confer increased viral fitness. So one of the key drivers is thus an uncontrolled viral transmission and COVID-19 infection. At the end of the day, if we don't have infections, the virus does not have the opportunity to replicate and mutate through natural selection. So it is key to drive viral transmission down. Transmission in people with partial immunity is also very worrisome because a variant that emerges in such patients will potentially be fitter because it has the opportunity to better adapt to our immune system. And that will provide the virus with greater fitness compared to other variants circulating at that time. Prolonged infection in immunocompromised patients, infections that take several months to be cleared out, might drive the selection of variants. And this has been observed that the virus can change quite dramatically in these situations, as is thought to have been the mechanism that led to the emergence of the UK alpha variant. And the other thing that not to forget is that a virus that goes from human to an animal may adapt to survive in an animal And then when it goes back to the human population, it might have increased potential for spread, immune evasion, and pathogenicity. So in these slides, we have a compilation of mutations that have arisen in SARS-CoV-2 spike due to sustained transmission. This is a slide from, this was compiled in January, so more have emerged meanwhile, but you see that mutations fall into 4 general classes. Those that affect the receptor binding domain, which are of importance because some may provide both immune escape and the fitness advantage. There are some that occur in the amino terminal or NTD domain in yellow, which is the portion most exposed of the virus surface. And here there is evidence for immune selection in this region and preliminary evidence that at least one of these changes could improve fitness. We also see mutations in or near the furin cleavage site, and several grouping close to the D614G mutation, possibly affecting infection efficiency and can also be important for neutralizing antibodies. But generally, what I want to point out is that we can see that there are numerous mutations that have already arisen, and that are many that have arisen in animals as well as in humans. So I talked briefly about prolonged infection in immunocompromised patients. But what I want to show you here is an example of how this mechanism can promote the emergence of variants. This study that has been published in the New England Journal of Medicine shows what happens during in vivo evolution. Here in Boston, an immunocompromised patient with COVID-19 had persistent infection that lasted for more than 5 months before dying. The patient was sampled at 4 different times and had their viruses sequenced. And they observed that the subimmune response, instead of clearing the virus, drove selection of viral populations with the same mutations observed in the recently identified variants. Now, of course, there are limits to what might happen during within-host evolution and on a prolonged infection, but it was quite surprising to see the emergence of the alpha variant that was quite genetically different than the viruses circulating in the UK at the time, with 23 different mutations associated with fast spread. So this is a cautionary tale about the importance of not only doing population-wide surveillance and sequencing, but also, besides of course helping clear the disease, also following the viral profiles of hospitalized patients for long periods of time, as they might be the breeding ground for selecting new variants. So recently we've been hearing about the Delta variant. So let's talk about how it emerged and what it means for the progression of the pandemic. Early in the year 2021, we observed how in India, many variants circulated carrying many of the mutations presented in the Delta variant. SARS-CoV-2 Delta variant, also known as lineage B.1.617.2, is a variant lineage of the B.1.617 lineage, and it was firstly detected in India in late 2020. This variant has mutations in the spike protein, including the E484Q and the L452R, which are known to affect transmissibility of the virus, as well as whether it can be neutralized by antibodies produced for or by previously circulating variants of the virus. As you can see in this graph in red, the B.1.617 grew faster than any other variant of concern circulating in the country, and is thought to be partially responsible for India's second wave of the pandemic beginning in February 2021. The B.1.617 is currently divided in 3 different subclades that have been spreading and have been detected in 98 countries around the world. From these subclades, the B.1.617.2 has been observed to have attack rates to be 50 to 70% higher than the alpha variant, which prompted the World Health Organization to classify the sublineage as a variant of concern and said that it showed evidence of higher transmissibility and reduced neutralization. So the Delta variant is currently the dominant variant in many countries around the world, including India, the UK, South Africa, and has recently become the dominant variant in the USA, rapidly replacing the alpha variant. Before we go into these graphs, there are important caveats about these, as they depend on the viral sequence data that is uploaded. So they are influenced by the number of sequences uploaded, but also if they are biased, for example, due to targeted sequencing, or if they represent well the diversity circulating across space and time. So all these effects will definitely influence interpretation. But as you may see in the top graph, in India, we see that when the alpha variant, or B.1.1.7, started circulating, it constituted a maximum of around 40% of sequences represented in public repositories. But we see a precipitous drop in the beginning of April, coinciding with the rise of the Delta variant, or B.1.617.2 lineage, that currently represents more than 80% of all variant sequences from India. In the US, in the bottom graph, after the emergence, excuse me, of the alpha variant in late 2020, it quickly became the dominant lineage, shown by the green line, accounting for more than 60% of all sequences available. The Beta variant and the New York variant of interest were also accountable for many cases. However, a bit after the emergence and introduction of the Delta variant in the country, it rapidly spread and rose in prevalence while driving the decrease in the frequency of other variants, and just last month, it became the dominant lineage in the USA. Studies have been investigating the ABCs of the Delta variants. These variants have mutations for which there is evidence that they confer a higher basic reproductive number, or R0. The R0 represents the average number of people a single infected individual can infect. And so it is a measure of how fast the virus transmits and spreads. The RT represented in the y-axis of this graph represents the average number of people a single infected individual can infect at a particular point in time. So in this graph, we see variant-specific estimates of RT using the US CDC case counts and GISAID sequence data, and it suggests an RT for the Delta variant of 1.18 averaged across 20 states analyzed, which is significantly higher than what we observe for other variants. So the rapid increase in frequency and dominance of the Delta variant can be explained by it being more transmissible, even more than the Alpha variant, but it also appears to be more immune evasive, possibly more than the Beta variant, as we see people being reinfected with this lineage. So in this table, we see vaccine efficacy estimates by variant. The spike protein, as I showed you earlier in the presentation, is the focus of most COVID-19 vaccines, as it is the part of the virus that enables it to enter our cells. We also know that no vaccination is perfect, and we've seen drops of different magnitudes for different vaccines in terms of preventing disease and infection with the Delta and other variants. So while we may see variants that make some vaccines less effective, we think it's unlikely one will emerge that completely evades our vaccines. Fortunately, as we see here, most vaccines still exhibit relatively high rates of protection against the circulating variants. So the population should continue to get vaccinated, as vaccines are still our best chance to control the spread of COVID-19 and the emergence of future variants. Nonetheless, given the impacted vaccine efficacy and increased transmissibility recorded for many of the variants, as well as the high infection rates in many regions of the world, herd immunity continues to be a moving target, suggesting that more people need to be vaccinated in order to achieve herd immunity. This means that we should continue and, if possible, increase vaccination efforts. Additionally, these viral characteristics point that there might be value in additional vaccine doses via boosters to increase protection levels. Disparities in vaccine distribution around the world pose another challenge in the control of the pandemic, as even if there are large sections of the population that are immune, there are also large sections that are still affected by the virus, where new variants can emerge and spread. So while we wait for vaccination, we should maintain social distancing measures and wear masks to help control the spread and prevent opportunities for the virus to evolve into something more dangerous. Ultimately, the rise of variants is a reminder that the pandemic isn't over, and so efforts must be made to stop replication everywhere. So to conclude, our top priority should be to generate data for public health agencies looking to provide real-time information. We knew that mutations and variants would come to be important, and so genetic information can be studied with multiple goals. The data generated also allows for continuous improvement and development of methods such as methods for sequencing, like the Arctic protocol, bioinformatic tools for analysis, or phylogenetic models to study the evolutionary and transmission dynamics of the virus. Besides the practical aspects of public health, it is important that this data is also integrated with a scientific ecosystem, including that it is analyzed by molecular and evolutionary biologists, immunologists, hosts, geneticists. And finally, it is important that teams around the world have the ability to produce this data. So it is crucial to support international efforts like those led by my team at NIH in collaboration with folks from the Johns Hopkins University Applied Physics Laboratory. We have always been committed to capacity building around the world in the fields of epidemiology and phylodynamics. But during the pandemic, our efforts were rapidly ramped up and tailored to assist laboratories around the world to be able to sequence and analyze SARS-CoV-2 genetic data in order to understand the viral transmission dynamics locally and thus better implement control strategies. In fact, the scale of what most countries sequenced before the pandemic and what they are doing now is quite extraordinary. Nonetheless, there is still high heterogeneity in the number of sequences produced around the world, with countries that have reported cases, but for which there are no viral sequences available, as you can see in the map in gray. Public health agencies should organize to scale up the number of samples sequenced. The samples selected should be representative of specimens across the country and over time. And it is also crucial to collect the related patient metadata, including information regarding the patient's travel history, vaccination status, and record of previous infections. It is important, of course, to sequence both hospitalized cases, but also randomly in the community to try to go across it to work out what's infecting the population. If possible, one can also direct their sequencing capabilities on much more targeted sequencing to investigate severe disease or the cause of vaccine failure or the cause of reinfections. It is also particularly interesting what people might be carrying when they come into the country, as well as enriching for variants of concern, so research testing. A sequencing capacity can also be interlinked with other studies, such as for vaccine trials or studies in care homes, prisons, etc., or to study the link between viral and host genomes. So overall, this information makes it possible to investigate how variants impact COVID-19 severity and the effectiveness of vaccines, treatments, and diagnostic tests. It also helps characterize the origins of an epidemic and how it spreads over time within the country or overseas. And thus, it gives us an insight into the viral transmission dynamics. And so I take this opportunity to welcome you to contact me in case you need assistance in sequencing SARS-CoV-2 or help analyze your own viral genetic datasets using phylogenetic or advanced phylodynamic models. Nonetheless, we have to be realistic that the road to control the COVID-19 pandemic is arduous. And even though we can take precautionary measures to avoid transmission, the science has to be done to determine the effectiveness of current therapeutics and vaccines against the newly detected variants, which helps assess their effectiveness to the public. But it takes time to produce evidence for us to take informed decisions on how to control and curb the pandemic. However, this is only possible if the data is being rapidly shared, because otherwise we can't get reliable and, most importantly, timely results which are needed to react in an evidence-based way to what's happening in the pandemic. So with this, I'd like to thank the immense number of scientists that I have worked on during the pandemic in collaboration or through training initiatives. And thank you also very much for listening, and I'd be happy to answer any of your questions.
Yeah, it's great presentation, Nadia, and all of the students and the participants learned very depth understanding about the virus of concerns and virus of importance, and specifically about how the concern of the delta variants and what are the emergence of that. So it's a really great presentations with the covering of the all aspect of the virulence in the importance of how this spread it's took out in the previous way and what are the concerns will be how to taken care for that. Okay, so we have the few questions, Nadia. I will go one by one.
Absolutely.
Yeah, just a minute. Okay, so one question is related to the vaccinations. How safe these vaccines for the children's like that? So any specific suggestions or your comment regarding, because you are the expert in this kind of the area, so everyone's know about this thing specifically.
Yeah, if I understood correctly, is how safe the vaccines are for children, correct?
Yeah, yeah.
Yeah, so the vaccines have been formulated indeed for the adult population, and trials initially were mainly done in adults. But while the vaccine was being deployed in the adult population, trials started to be performed for children, for adolescents first, teenagers, and then children. And currently, many vaccines are already approved to be deployed in the younger population, so they are definitely safe.
Okay, that's great. So no worries about this.
No. Everyone should get vaccinated, even the younger populations, if possible.
Yeah, perfectly fine. The second questions from the Ali Samay. how about the epsilon mutant or variant? Might be I not correctly read this because the question is little bit tricky on the part. So, but what I understand that he asked about the how these mutations will be the often in the terms of this any kind of the model we can suggest on that.
The epsilon variant is, let me just do an escape so I can also see the questions. But as I was mentioning, the epsilon variant is a variant of interest. And even though it has mutations that might confer either increased transmissibility or a slight immune evasion, it hasn't yet been scaled up, as I mentioned, to a variant of concern. So we are monitoring it to see if it starts rapidly spreading in the population. And if that is the case and starts to have an impact in the control of the pandemic, it will definitely be scaled up to a variant of concern.
Okay. The another questions like that, can existing vaccines work against the Delta strain? I think you already saw this in the slide, but can you light some on these questions?
Yeah. So the vaccines work with the beta strain, even though the beta strain has mutations that cause some level of immune evasion. Even though the degree of protection is slightly lower, it's still quite high, even compared to any other vaccines that one takes, like the influenza vaccine and others. So absolutely, even in places where the beta variant is the dominant, one should still be vaccinated, as it will confer some protection to COVID.
The same questions on the Delta variant severity. So someone asked that, can we predict the Delta variant severities like that, or how the severe this variant would be in the when the virus enter in the human body? What kind of the severe things or damage things can be happen by after that? Any kind of the severity you can predict on that?
Absolutely. So I will not predict, because we already have some preliminary evidence that the Delta variant is indeed more severe than the previous variants of concern. It is more transmissible, so it takes less exposure to get infected by the Delta variant. It evades our immune system, so our antibodies cannot clear the virus. We have seen some more severe disease in the populations that are infected with these variants. So it is indeed more severe than the others.
Okay, great. There is a one interesting question someone asked that, is any possibility to say that the SARS-CoV-2 entered through the non-SE routes like the receptor? Maybe there is any other route will be there or entering this virus?
I'm sure that there are other alternative receptors that could enable infection from, but the main receptor that is used in viral infection of the cell is the ACE2 receptor.
Okay, great. The one questions from the students, basically he would like to know that what should be the targets of medicines to identify and destroy the virus? Can we use the gene modifications to reduce the mutations rate? Okay. So can you light on some?
Yeah, of course. So the medicines to identify and destroy the virus, I wouldn't say that gene modification is the easiest way to go about it. First of all, we should prevent infection. And to do that is really wear masks, get vaccinated, and social distance. The other way is, once one gets vaccinated, if available, one could be treated with convalescent plasma that has already antibodies from previously infected patients that recovered, and that would help identify, because those are antibodies, and destroy the virus. Vaccines, of course, are a good way, again, because it builds these antibodies in our own bodies. gene modification could help, but it's such a complicated biological experiment to do. And also deploying a virus that has a modification in its genes might be somewhat controversial, since we already have variants that are quite dangerous. So I would be cautious on doing that.
Yeah, great. So that is a very one nature. Okay, so the question is regarding the what is the rate of the mutations of the COVID-19? Any baseline or any kind of the standardized baseline for that, like the variant of emergence or variants of importance for the COVID-19?
Yeah. So SARS-CoV-2 is an RNA virus, which usually one would expect that evolves quite high, to the 10 to the minus 3 substitutions per site per year. But this RNA virus has a proofreading mechanism, which makes it to have an evolutionary rate that is slightly lower at a baseline of 10 to the minus 4. However, as I showed you in the slides, these recent variants tend to evolve slightly faster than the baseline rate of 10 to the minus 4, and consistent with the evolutionary rates of influenza viruses at 10 to the minus 3.
Great. So there is a questions regarding the booster dose. So I just rephrase these questions. Do we need the booster dose of the vaccines? That's a very simple one.
So for now, I guess our priority is to just get the first dose, right? The first dose if the vaccine only has 1 dose, or 2 doses if that's the vaccine composition. But of course, in the future, since these vaccines were produced for the earlier COVID variants, perhaps it might be helpful to develop vaccines updated with the current variants. And those would be a booster dose of vaccine that would allow us to be even more protected to the current circulating variants.
Okay, great. Is any fungal or like the correlation between the viral infections and fungal infection? Do you find any kind of a sequence evidence regarding that, or there is one question?
Sorry, Curtin, could you please repeat?
Yeah. The questions regarding, did you find any evidence on the correlations between the viral or fungal infections in the terms of the sequence, both of like the viral sequence and the fungal sequence? Is any kind of the correlations in the both the infections? Because
so correlations of infections in the sequence of viruses and hosts.
Yeah, fungal infections. Okay. Yeah, I think.
Fungal infections. I'm sorry, yeah. I was couldn't understand. So I haven't studied in detail fungal infections and COVID simultaneously, so I haven't seen any correlation between mutations in both the viral and fungal infections. But what I've seen is that the fungal infections really contribute to more severe disease when in combination with COVID-19.
Okay, great. The last questions, does Delta virus affect the brain tissue? Since the brain is also has the ACE receptor, so how can we affect the patients when is compared to the Alpha variant and in the brain tissue particularly? So it's specifically on the Delta variant and that severity on the brain tissue part.
That's a great question. We indeed have seen some preliminary patients, particularly children, that tend to be affected. Their neural tissues are affected by the Delta variants, and that might have consequences if we are not able to curb the infection in children, as there might be sequelae in the neural tissue. There is indeed some ACE receptors in the brain. And we've seen more of these effects indeed in infections caused by the Delta variant compared with other variants of concern.
Okay, so further of these questions, see the Priyam also write that give the increasing rate of the evolution of the virus, as you mentioned that the R rate is very highly in that case. So how likely we to tackle more advised variant, what is the best method to keep up with those variant, and how long would we take in the overcome with the evolution together? Means the concern about the concern about that, how we can tackle these viral evolutions. Can we simultaneously with our evolutions that both are should be there fine, or it's okay with that?
That's again another great question. And as I mentioned before, ultimately, if there are no infections, the virus does not have the opportunity to evolve and mutate into something more dangerous. So our goal should be to curb transmission of the virus. And that can be done by simple measures like masks and social distancing, and of course, ultimately by vaccination. That will be the way to curb the pandemic. Of course, there are also therapeutics that have been that are under development, but the goal is to, of course, curb transmission dynamics, so infections, to avoid having to use these therapeutics that might add some pressure to the virus and even select for more dangerous variants.
Great. Thank you, Nadia, to answer all of the questions, and I hope everyone is enjoy your talk with the very great manner. And so we are ending of the sessions of the second session. So I would like to thank the GSFs University to hosting such a beautiful webinar and provide the platform for this particular expert to discuss about this particular talk. Specifically, our Vice Chancellor, Professor Dr. Nikhil Javeri, Director of Administration Sri Panjal sir, and the Dolakia sir. So, and all the participants and specifically colleagues of my biotech department who are helping me to organize such a beautiful webinar series. So thank you all. Again, I welcome if any kind of the questions, you can send the mail. I already mentioned in the particularly webinar mail address, webinar.gsfcac.in. And thank you. So thank you, Nadia, for
thank you very much for having me.
Yeah, thank you. So, and once again, the certificates of this webinar will be distributed accordingly or the feedback form, and the timing of the particular sessions joining both the sessions will be counted as a certification for that. So have a good day, everyone, and hope we are seeing the upcoming days with the different seminar series. Thank you, Nadia.
Bye-bye. Have a great day.